Good morning, students. Today we are coming to discuss about one of the most important structures in the eye, known as the cornea. We will describe its gross anatomy, enter into histology, talk about its nerve supply, its functions, and so on and so forth. Just to set the ball rolling, this is the cornea. Okay, it has this anterior curvature. This client is lying down and looking at the ceiling. All right, so this is the cornea. It starts from the limbus here. Sorry, it starts from the limbus, comes, it has anterior curvature like this, and ends here. It is transparent, as you can see. All right, it is transparent. So, um, it measures horizontally 12 millimeters. Okay, this horizontal diameter is 12 millimeters and its vertical diameter is 11.5 millimeters. That is anteriorly. In its posterior curvature, it measures 12 millimeters in the directions, but externally it measures 12 millimeters horizontally and 11.5 millimeters vertically, because superiorly and inferiorly, it has slightly been invaded by the conjunctiva, covering its superior and inferior portions mildly. Centrally, its thickness is 0.5 millimeters. 0.5 millimeters. But at the periphery, its thickness is 1 millimeter. Let's talk about its microscopy. Okay, microscopic anatomy. The cornea has 5 major layers, alright? And these layers, so this is the cornea as you see here, alright? And it has a better appearance in the next. So this is the same structure over here, okay? Let's talk about its layers microscopically. It has 5 major layers, alright? So these include the epithelium, as you can see here, epithelium, that's the most external layer, alright? Followed by the Bowman's layer. Look at the Bowman's layer here, alright? That's the Bowman's layer or Bowman's membrane. Followed by the stroma. As you can see, this is the stroma of the cornea, alright? That's the stroma. From the stroma, the next in line is Descemet's membrane. Descemet's membrane, okay? Descemet's membrane. This Descemet's membrane, as you can see here. The last but not the least, of course, is the endothelium, the cornea endothelium, okay? Cornea endothelium, which has a touch with the aqueous humor. Aqueous humor is here, and that is your endothelium. Now, we'll describe few things about each of these layers for better understanding. Okay, so all epithelia in the body have their description, and that of the cornea is not an exception. So the epithelium of the cornea is stratified. When I say stratified, it means that it has layers, doesn't it? Yes, it has 5 layers. Okay, so well, the cornea epithelium has 5 to 6 layers of cells. All right, so that is the meaning of stratified. Stratified means it has layers. So it is stratified. It's also squamous. When we say squamous, it appears like scale, okay, of fish, and it has that resistance. Yes, so it is stratified, squamous. When we talk about keratin, keratin is more or less the structure, the substance that the pores of four-legged animals like lions, cats, horses paw. What they have, we call it keratin. Does it resemble it? No. So it is non-keratinized. Does it secrete? No, it doesn't secrete. So it's non-secretory. So in a summary, The epithelium of the cornea is stratified, squamous, non-keratinized, and non-secretory. Stratified, squamous, non-keratinized, non-secretory epithelium. And this epithelium has about 5 to 6 layers. Okay, all right. Now there are few cells that it's rich in. Notable among them are surface cells. Okay, surface cells are most found at the superficial portion of the epithelium. Then we have basal cells. The basal cells are found at the base of the epithelium, okay, getting to the junction of Bowman's membrane. Other cells are called the wing cells. The wing cells are at the wings, okay, of the epithelium. The last but not the least cells are limbal stem cells. The limbal stem cells within the epithelium. So these are the four cardinal cells which the epithelium has. Now, the surface cells, what do they do? Almost every 5 to 6 to 7 days, these cells they shed off into the tears and are gone, and they are replenished through mitosis with new cells. Divisions that may come from where? The wing cells and then the basal cells. Okay? Alright. It is also important to note that the epithelium has a basement membrane. Okay? It has a basement membrane. We should also note that the surface cells of epithelium is rich in certain microscopic structures known as microvilli and microplici. Microvilli and microplici. These look like small protrusions from the epithelial surface. What do? They help in accommodating the tear film. They create spaces where the tear film can enter, including the mucin. Alright, they can enter and deposit themselves. Okay, so that is that. Now, let us focus on so let's focus our attention on the Bowman's layer. Okay, this is the Bowman's layer. For your information, among all the 5 layers that the cornea has, it's only 2 layers that once damaged, they can regenerate. And these include one, the epithelium, alright, the epithelium, because one, the surface cells they are replenished, two divisions that come from the ring cells and also the limbal stem cells. Two, the cement membrane, okay, because any loss from the cement membrane is replenished from or by the endothelial cells. So these are the only 2 layers that once damaged they can regenerate. The rest of the layers, once they get damaged, the cornea becomes scarred because there's no regeneration. So talking about the Bowman's layer, as I've said, incapable of regenerating once they get damaged. Okay, let's focus our attention on the stroma. Okay, the stroma of the cornea. The stroma is also known as what? Substantia propria. The stroma is known as substantia propria. Alright, so the stroma is rich, it constitutes 90% of the thickness of the whole cornea. Okay, the stroma constitutes 90% of the thickness of the whole cornea. And interestingly, 80% okay of the stroma is made up of what? Water. So 90% of stroma, the whole cornea thickness, 90% of it is made up of the stroma. And it's also all its structures, it is rich in 80% okay of water. And it's the rest of the 20% is made up of what? glycosaminoglycans, okay, found in extracellular matrix. So the rest of the 20% is made up of what? Glycosaminoglycans. Glycosaminoglycans are type of carbohydrates, okay. So there are three major fractions of the glycosaminoglycans, all right. 50% of them is made up of what? Keratan sulfate. Keratan sulfate. And that 25% is made up from chondroitin sulfate. And another 25% is made up of chondroitin phosphate. So 50% keratan sulfate, 25% chondroitin sulfate, and 25% chondroitin phosphate. That is the stroma and its composition of the glycosaminoglycans. Let's talk now about the Semmet membrane. Okay, the Semmet membrane. Remember, this is moving from outer part of the cornea into the inner part. Okay, so just after the stroma, what do we have? We have the Semmet membrane. It constitutes, as you can see, the Semmet membrane. It constitutes basement membrane of the endothelium. Okay, it is the basement membrane of what? The endothelium. All right. So it is secreted and regenerated by endothelial cells. So the cement membrane is secreted and regenerated by endothelial cells. Okay, it is secreted and regenerated by what? Endothelial cells. In that same manner, when there is damage in the cement membrane, okay, the corneal endothelial cells are able to reorganize themselves and reform the cement membrane. Because in a nutshell, the cement membrane, okay, it's what? Secreted and regenerated by endothelial cells. It's rich in type 4 collagen fibers, okay, and these are another important thing is that it helps information of the iridocorneal angle, all right, ending in what we call the Schwabi's line. It terminates in the Schwabi's line. That will be at the iridocorneal angle, the Schwabi's line. Now let's draw attention to the corneal endothelium, right? So that is the corneal endothelium, okay? That's the corneal endothelium. So it is a single layer of polygonal and cuboidal cells. A single layer of what? Polygonal and cuboidal cells. Another important thing that you should know about the endothelial layer is that it has tight junctions. Students, remember something. All this place is made up of aqueous humor. It's made up of aqueous humor, water. So if it doesn't have tight junctions, to regulate how much water goes into the other layers of the cornea, the whole cornea will be imbibed with a lot of water, it will be edematous, and it will let our vision be what? Blurred. So there is a tight junction. What does it do? It regulates how much water goes into the other layers of the cornea. So this is how it does it. If the cornea becomes dry, the tight junction opens up a bit more for aqueous humor to go into the other layers of the cornea to rehydrate. But if the cornea has a bit more water than it should, then there's a, we call it endothelial pump. Endothelial pump. It pumps water from the other layers of the cornea, especially from the stroma, which is 80% water. It pumps water back into the aqueous humor to let the cornea have its normal consistency and clarity. So, endothelium does not regenerate once it gets damaged. It has microvilli, okay? Microvilli. And we should know also that the corneal endothelial cells are about 2,500 cells per millimeter square. Per each millimeter square, you have 2,500 endothelial cells. And every year of our life, we lose 0.6% of this population. Each year of our life, we lose 0.6%. That cavity which is left because the endothelial cells are lost over there, the neighboring endothelial cells enlarge to occupy the space that has been left over by the dead ones. And this process through which the neighboring endothelial cells enlarge to occupy the space which is left by other ones that have died, we call it sliding. We call it sliding. It is called what? Sliding. Okay? So, when the endothelial cells reach a population of 500 per millimeter square, the cornea becomes edematous because now it cannot regulate how much water goes into the cornea, how much aqueous humor goes into the cornea. A lot of it, because the tight junction is lost, a lot of aqueous humor enters the cornea. The cornea becomes edematous and our vision becomes blurred. Having spoken about the layers of the cornea, the next thing to discuss is the functions of the cornea. Functions of the cornea. What are the functions of the cornea? Okay, there are three major functions of the cornea. One, it helps in light transmission. There are different wavelengths of light. Specifically, the light whose wavelength is between 400 to 700 nanometers. In a nutshell, these are the range of light that is safe for our sight. So 400 to 700 nanometers of light. Okay. So one, it helps in light transmission. Two, it helps in light refraction. Okay. It helps in light refraction. So what is light refraction? When light from the environment enters, the consistency of air in the environment is not the same as the consistency of the cornea. So once light passes from the air onto the cornea, it may suffer a deviation. That's what we call refraction. Okay? So it helps in light transmission, it helps in light refraction. Alright? The third function is that it helps in protection. Remember the concentric layers of the eyeball. We said that the sclera is the most external structure, and that in continuation of the sclera we have the cornea, and they are there to protect the other layers, the other concentric layers of the cornea. Going back to light refraction, okay, remember that the total refractive power of the eyeball is 60 diopters, and the cornea has 40 to 43 diopters. In other words, about 70% of the power of the eyeball lies with the cornea. And talking about refraction, refractive index of the cornea is 1.37. 1.37. Okay? Now, let's talk about metabolism. For every tissue to survive in the body, two substances are essential. These are one, glucose, and two, oxygen. There is no living structure which can live without glucose and oxygen. So, cornea metabolism. Let's talk about the energy that it needs. Okay, basically how it gets its glucose and its what? Oxygen for survival. So, sources. Alright. The cornea look at it this way, all right? It has two major sources. One comes from the inner portion. In that case, the posterior half of the stroma plus the descemet membrane and endothelium, they benefit from the aqueous humor. Whereas the anterior half of the stroma, basically the Bowman's layer and epithelium, may benefit from the precorneal tail film and other structures. Alright, so talking about how the cornea gets its glucose. Number one, it gets glucose from the aqueous humor. Okay, it's clear this is the aqueous humor area. So it gets glucose from the aqueous humor. Number two, it gets it from what? The precorneal tail film. Okay, it gets glucose from where? The precorneal tail film. Alright. Again, there are perilimbar vessels. The junction between the cornea and the sclera or conjunctiva is known as the limbus. There are certain vessels, okay, that are found at the limbus. So these vessels carry blood, and the blood will always be rich in glucose. So another source is what? The perilimbar capillaries. Okay, glucose may also be stored in form of what? Glycogen. Okay, glucose might be stored in form of glycogen in the corneal epithelium. Okay, when there's excess glucose in the cornea, it is stored in the corneal epithelium in form of what? Glycogen. So when the need arises, this glycogen is broken down through process called glycolysis, okay, to release more glucose onto the cornea for use. Again, through Krebs cycle, okay, through Krebs cycle. Remember, the perilimbar vasculature carry blood, and through Krebs cycle, we can also get energy or glucose in form of what? Adenosine triphosphate. So these are the sources of what? Glucose. So I mentioned again, aqueous humor here. All right. We also have the precorneal tail film. Okay. Again, we have perilimbar vessels. Okay. Again, glycogen which is stored in the epithelial cells here. All right. Another source is where? When this glycogen stores break down through glycolysis, they release the glucose for the use of the cornea. Another source is what? Through Krebs cycle that may happen at the level of the perilimbar vessels, releasing glucose in form of energy, in form of ATP, which is adenosine triphosphate. So these are the sources of glucose that serve the cornea. Now, let's talk about sources of oxygen. Students, remember that the aqueous humor is rich in water, and water, the formula is H2O, at least 2 atoms of hydrogen and 1 atom of what? Oxygen. So, this tells you that the aqueous humor is rich in oxygen, alright? So, endothelium acquires oxygen from aqueous humor. Of course, not only the endothelium, but as I said, the posterior half of the cornea may gain from the aqueous humor, but specifically in this case, what will it gain? Oxygen. All right. And from the anterior half, what do we have? The epithelium acquires oxygen from the what? The perilimbar capillaries. Okay, and of course the precorneal tail film. Okay, so the precorneal tail film here. All right, and the perilimbar capillaries, they give oxygen, okay, to the anterior half. And posterior half, what do we have? From the aqueous humor principally. So let's talk about how the cornea is able to maintain its transparency, because it's supposed to be transparent all the time. Okay, so the cornea is transparent because of several factors. One of them is that it's as a result of its relative dehydration. Okay, the cornea is relatively what? Dry. Why is it relatively dry? Simple. The third junction of the endothelial cells here, they regulate how much water enters, okay, to hydrate. Just enough water enters, because if it is edematous, it will obscure vision. Number two, it is transparent because it does not have cell, and it doesn't have blood vessels to start with. It doesn't have pigments, and relatively it has fewer cells. Okay, so why is it transparent? Another factor is what? Acellularity, relative acellularity, doesn't have blood vessels and doesn't have pigments. Another factor is what? It has a regular metric structure of corneal fibrils, especially within the stroma. They are regularly what? Arranged. Okay, then it also has a consistent refractive index of all layers, which is 1.37. All right. So these are the four major reasons why the cornea is transparent. One, let me repeat: relative dehydration. Two, relative acellularity, lack of blood vessels and pigments. Three, regular matrix structure of corneal fibrils. Four, consistent refractive index of all layers. All right. So these are the reasons why the cornea is what? Transparent. I may further tell you that the relative dehydration of the cornea is due to what? Anatomic integrity of endothelium and epithelium. So the epithelium will not let too much water enter your cornea. Assuming you are swimming and your head is always in the water, okay? The endothelium also has some tight junctions which regulate how much water enters. So because you are in water for a long time does not mean that a lot of water can enter your cornea through the epithelium. Okay, so anatomic integrity of epithelium and endothelial cells. Okay, another reason is that the endothelial pump removes fluids from the stroma. So assuming that there is so much water in the stroma, the endothelial cells will help pump out all the excessive water in the stroma back into the aqueous humor. Another reason why the cornea is dehydrated all the time is that there is evaporation of water from the tear film, okay? And this increases osmolarity of the tear, drawing water from the cornea. So let me repeat this particular portion. So because the cornea epithelium is exposed to the environment, okay, the precorneal tear film can easily dry, and when it dries, it leaves the sodium, potassium, the chloride, all the substances that I told you the aqueous layer of the precorneal tear film has. Okay, it leaves these solids, creating osmolarity, and by physiology, water will be drawn from the cornea, okay, towards where? It draws water from the cornea stroma towards the epithelium, and in that order the cornea becomes what? Dry. The last but not the least, to explain why there is relative corneal dehydration, is known as what? The normal intraocular pressure. Okay, So far as the intraocular pressure in the eye is normal, the cornea will always be dry, relatively dry. Okay? So, having spoken about the factors, I repeat, which include why the cornea should be transparent, there are four. Okay? Relative dehydration, relative acellularity, lack of blood vessels and pigments. We also have regular matrix structure of corneal fibrils. The last but not the least is what? The consistent refractive index of all layers, which is 1.37. Okay, let's talk about the innervation of the cornea. The cornea gets its nerve. For your information, it is the most sensitive structure in all parts of the body. All right, it has a lot of nerves. It's rich in sensory nerves. So to start with, we have the trigeminal nerve, a branch of the trigeminal nerve known as the ophthalmic branch, and a branch of the ophthalmic branch also has three major branches. These are the frontal nerve, the lacrimal nerve, and the nasociliary nerve. Out of this, let's narrow down our discussion to nasociliary nerve. Okay, the nasociliary nerve also has certain branches. Okay, one of them is what? The long posterior ciliary nerve. So nasociliary branch also has one major branch, which is what? The long posterior ciliary nerve. Long posterior ciliary nerve. And this long posterior ciliary nerve also has three major branches. These are one, the annular plexus. What does the annular plexus do? The annular plexus serves the limbus. Okay, it gives sensation to the lumbar region. Another branch is known as what? The intraepithelial plexus. Intraepithelial plexus. What does it do? It gives sensation to the corneal epithelial cells or epithelial layer. Okay, it gives sensation to the corneal epithelial cells or the epithelial layer, as you can see here. The last but not the least is the subepithelial plexus. Okay, subepithelial plexus. It gives sensation to the other layers that are found beneath the epithelium. So we can talk about the Bowman's, stroma, the deciments, and of course the endothelium. Students, our discussion has been quite long. Go over several times, and I know you are going to understand. If you have any difficulties, put them down, and once we meet in class, we are going to discuss further about it. So for your information, that is the cornea, and never forget about the cornea. In your daily life, read, go over the slides, the YouTube several times, so that you can get better understanding. If you just do it once, it will not take. So until I meet you again, learn very hard.